Hello everyone, my name is Prarthana Guha. I'm a PhD student at the Department of Chemistry and Biochemistry. My poster is on roles of IDO1 in inflammation and macrophage activation. What is IDO1? IDO1 is one of the three major enzymes in the tryptophan catabolizing pathway. The role of IDO1 is to convert L-tryptophan into kynurenine, which is the rate-limiting step in the whole catabolizing pathway. Now, ID1 has its effects through several effector mediators such as AHR, GCN2, mTOR, etc. Why is ID1 so important? Because it has been shown that ID1 has many significant effects on immunosuppression, inflammation, gut microbiome, and cancer. Now, my objective is to determine the role of ID1 in macrophage activation during inflammation and also study the regulation of inflammatory cytokine expression in macrophages, whereby I want to get some good therapeutic targets for future study. Now, for this study, I have used DHP1 macrophages, which are human macrophages, and raw mouse macrophages, where I have treated them with lipopolysaccharide, a material of gram-negative bacteria, where it acts as the inflammatory stimuli. And now I have got the RNA from that, and using the RNA, I have run quantitative real-time PCR to see how much RNA expression varies among each other. Now, if you look at the left panel, you'll see that the first step of the of any macrophage human macrophage study is to convert the monocytes into macrophages. Now, there are some markers involved in the depiction of differentiation of macrophage, and I have used DAPI-FITC to determine that, and you can clearly see that there are expressed macrophages. That means the PMA treatment we have given has turned our monocytes into macrophages. Now, if you look at the panel number 2, I have used LPS to treat those macrophages, and you can see that there is an elevated level of IL-6 and IL-1 beta, which are pro-inflammatory cytokines, depicting that the inflammation has worked. And also under this inflammation condition, the ID1 level is up. Now, the similar result is shown in the mouse macrophage, where IL-6 level is up and hot air level is up. So this is to determine that LPS is a good inflammatory stimulant. So whatever data we have on IDO1, it is upon LPS treatment, and IDO1 is upregulated during LPS treatment. Now, NF-kappa B is a regulator of this inflammation process during innate immunity. So the next step we did is to inhibit this NF-kappa B using a small molecule and see its effect on different genes. So upon the inhibition of NF-kappa B, which is a major regulator of innate immunity, we can see that some of the principles pro-inflammatory cytokines, IL-6, IL-1 beta, and our long non-coding RNA, which our lab specializes on, is also down upon the inhibition. Surprisingly, what we saw is that IDO1 is also down upon NF-kappa B inhibition. That means IDO1 is, though it is treated with LPS, upon NF-kappa B inhibition, it goes down, which means NF-kappa B regulates the expression of IDO1. Further study is required on that. However, we can definitively say that IDO1 is induced upon treatment with LPS, and our preliminary data suggests that macrophages differentiated from monocytes, and when activated, they accept LPS as a good inflammatory stimuli, and upon those LPS treatment, some of the major pro-inflammatory cytokines are elevated. Our lab specializes on hot air, which is a long non-coding RNA, which has been long associated with inflammation. So this further proves that whatever inflammation we use has worked on IDO1, and IDO1 is upregulated. So hence, working on IDO1 and depicting its role in inflammation would be beneficial for therapeutic purposes. For this study, I want to thank all of my lab members, my principal investigator, and I also want to thank NIH for funding this study. Thank you.